We have looked on the data from the ADAPT trial. It was a large phase 3 trial performed in Germany at about 80 centers several years ago. And in this trial, so the major difference to other trials in early hormone receptor positive HER2 negative breast cancer was that we have combined static assessment by recurrence score, by genomic assay, with a functional test by Ki-67 response to preoperative endocrine treatment given for 2 to 4 weeks before the surgery. And this was done in patients with clinical intermediate to high risk disease. And after this risk assessment, patients were allocated to endocrine therapy or to chemotherapy based on this three-step approach, based on clinical assessment and genomic risk and response to endocrine treatment. And now we have looked on the prognostic assessment from this trial, and we have shown that use of genomic assay like Oncotype DX may be useful for the prognostic assessment to be done combined with clinical markers, for example, tumor size, nodal stage, or also expression of progesterone receptors on the immunohistochemical level. So it's not simply the genomic signature, it's also additional factors which may be used there. And one novel thing which was shown in our analysis was that in patients with high risk, so treated by chemotherapy in the trial, we know that patients with invasive lobular breast cancer have rather more favorable biology, but there is a small group of these patients with high risk features, and we have shown for the first time that this small group with high risk features may be treated very differently and very careful, because this patient has real increased risk for relapse. This is something new. So not to, we have to take histology also into account to understand how many residual risk will have this patient, for example, after chemotherapy. Yeah? And this is something which may be used also in the clinical routine, also on Monday next week. I think these results point out that we have to take into account, as well as histology subtype, and also, tumor biological factors, for example, progesterone receptor expression and genomic signature, and clinical factors, for example, tumor size or nodal status. All these factors should be taken together into account to have the more precise information for our patients for their residual risk after chemotherapy and standard management.